Various mechanisms that turn the immune system on. Okay, the communication between cells. We discussed antigens, we discussed antibodies, we discussed the role of T cell and B cell antigen presentation. All of this is to achieve what? Is to try to get rid or destroy a perceived threat. Now, I emphasize perceived because, as we will see, and as I told you several times, the immune system doesn't know what's good or bad for you, just what belongs and doesn't belong. So some of these mechanisms that I will discuss that are designed to protect you from something that could be potentially harmful, in certain cases actually act against the host, against your own. Okay? And anybody who has asthma or who has rheumatoid arthritis or who has lupus and other, type of situations where inflammation or the immune system is the culprit can understand that. And we cover that when we talk about the pathology of the immune system. So, effector mechanisms simply are the weaponry that the immune system uses to fight a particular perceived threat. Okay, once the immune system is activated against a particular threat, what does it do to get rid of it? Okay, and these are the effector mechanisms. Now, I want to emphasize, and I show you a particular slide to tell you, even though this is funny, that as small as these cells and molecules may be, they can be extremely powerful. So don't underestimate, okay? I mean, if you ever have either encounter or heard of toxic shock syndrome or systemic anaphylaxis, okay, when somebody you may have one of your patients die on you, okay? So these are very, very powerful entities. In most cases, they're mild, but in some cases, they're very severe. So when we talk about effector mechanism, what would we talk about? Well, in general, mechanistically, we divide the immune responses into humoral immunity and cell-mediated immunity. Now, I have to emphasize that these are divided for conceptual purposes, to make understanding of the immune system better, but they're not separate entities, and as a matter of fact, they're interconnected, okay? In fact, cell-mediated immunity is kind of a misnomer because all immunity is cell-mediated. Antibodies are made by cells. Cytokines are made by cells. So mechanistically, when we talk about humoral immunity, we talk about the soluble components of the immune system, antibodies, cytokines, complement. The reason why it's called humoral is because in the early days, when these molecules were started to be studied, they were found, of course, in body fluids, and because body fluids at the time were called humors, this was historically assigned the name humoral immunity. And the basic concept of humoral immunity that we'll discuss is antibodies and an antibody response, a humoral response, and complement. Okay? Now, I don't touch too much on cytokines because unfortunately we have a limited time to cover immunology in this course. Okay? We give 2 hours of cytokines to our students, just on those because they're very important. But I'll skim by them. So, basically, why is it that antibodies are important? Okay? Well, once antibodies are involved in recognizing or in attaching to a particular antigen, then what they do is to do many different things. Okay? For example, some antibodies, such as IgG and IgM, can activate the complement cascade. We'll talk about complement in a few slides. Or they can opsonize, and once again, opsonization is basically the coating of a particular particle, for example, a bacterium, with opsonin, such as antibodies and or complement components, so that particular particle is more visible chemically to the phagocytic system. They can also neutralize toxin. An antibody can attach to a toxin and either changes three-dimensional shape in space, so it becomes inactive, or actually affect the active side of the toxin. In case of IgA and secretory IgA, they can prevent colonization of mucous membrane. Why do you want to do that? Because many, many pathogens enters via the mucosa. Think of your respiratory system, think of your digestive system. And of course, for a pathogen to infect, it has to first colonize. For it to colonize, it has to first attach. So the antibody that can prevent attachment, prevents colonization and therefore infection. And of course, they can turn on various different accessory cells. Once the antibody is bound to an antigen, many cells can see that antibody on that antigen and act upon it. Okay? So do a lot of different roles. Now, humoral responses are characterized by different features. Okay? You have a primary immune response and a secondary immune response. The primary immune response usually relates to the humoral response that is generated when an antigen has been seen for the first time. On the other hand, a secondary response refers to the response to an antigen that it has been seen before. Okay? And there are differences between the two. For instance, the secondary response is enhanced. I'll tell you in a minute why. And also, a humoral response can pick and choose immunoglobulin classes or isotype. You can switch isotype. A cell that makes an antibody to antigen X can only make antibodies to antigen X, but it can change isotype going from IgM to IgG, for example, or from IgM to IgE, depending on the need. Why? Because as we've seen in the immunoglobulin lecture, they have different functions. Okay? So a cell can pick and choose which type of antibody class is more appropriate for their response. And the secondary response also has a high affinity than the primary. Okay? And the secondary response involves the development of memory. So here are the features of a primary response. Primary response usually involves a naive B cell. Naive B cell is a B cell that has never seen that particular antigen before, and therefore has not been preserved as a memory cell. Remember, I talked about saving the key that fits into the lock and put it in a pocket as a memory key? So it's a naive B cell. The lag period between exposure and reaction is longer than the secondary. Okay? The time between exposure and reaction in a primary is longer than the timing between exposure and reaction in the secondary. And as a matter of fact, this is the whole essence of developing vaccines. Okay? The concept of a vaccine is to fool the immune system to think that it has seen the particular pathogen before, even though it has not. So when the real pathogen comes in, it mounts a secondary response to the pathogen. Okay? It's really beating the pathogen to the draw. Okay? So for example, let's say in an imaginary simplistic situation, a pathogen kills you in 5 days. If it's a primary response and you take 7 days to respond, the bug wins. If the response is secondary, and let's say it only takes 3 days, then you win. Okay? So the use of vaccines, again in a very simplistic way, is to exploit these difference in lag periods. The magnitude of the response is lower than secondary, by that meaning how much antibody titers within the serum are generated to a particular antigen. Now the isotype, the type of immunoglobulin that is characteristic of a primary response, tends to be IgM. Okay? On the other hand, a secondary response involves the activation of a memory B cell. A memory B cell that has seen that antigen before and now it's prepared. Okay? That has been set aside so that if that antigen comes again, you're ready for it. The lag period is much shorter. In other words, the period between exposure and reaction is shorter than the primary. The magnitude of the response is higher, meaning the higher titer you make more antibodies. The isotype is predominantly IgG, okay, unless it's a mucosal immune response. And this involves the activation of T cells. So these are the difference between a primary and a secondary humoral response. Now, each response is antigen specific. So for example, let's say that you have an imaginary antigen A and an imaginary antigen B. And you inoculate, let's say, a laboratory animal with antigen A. The animal makes a primary response to antigen A. Now let's say that several weeks after this, you add a mixture of antigen A and antigen B to the animal, or to the host, and now look at what happened. You have a primary response against antigen B. Why? Because B has seen for the first time, but the response to antigen A is secondary, is much higher, much quicker. Okay? So each antigen can create its own response. And of course, this in turn complexes the system, because now you have the creation of a memory B cell. In other words, this particular B cell that was exposed to the first antigen, now instead of dying off, it's kept aside. In fact, several of these cells are kept aside, they're kept in circulation. So when that LOX comes back in, you don't have to fumble with a whole bunch of keys trying to see which one works. You have them in a separate pocket. You have them as memory cells, and of course then the secondary response is much higher. Now the isotype switch is a switch of types of classes of immunoglobulins. Remember in the first lecture we talked about different classes do different things. In a secondary response, the isotype can be switched to fit the situation. Okay? There's a whole complex series of gene rearrangement and genes a deletion in the constant region of the immunoglobulin that changes the isotype. I'm not going to go through the molecular biology of this, but just know that usually it's unidirectional from IgM to other isotypes. Okay? And this involves the collaboration between helper T cells and B cells. So B cells, which react to an antigen, okay, present to T cells, and then receive feedback signals from the T cell, and then and only then they can change isotype of their antibodies to that particular antigen. The other thing that happens in the secondary response is that the affinity of that antibody to that antigen is higher than the primary. The affinity meaning how strongly that antibody binds the antigen. The reason is that because the specificity of antibodies on a B cell is created randomly, you could have surface immunoglobulins to that antigen that are of high affinity, they're strongly bind the antigen, and surface immunoglobulin on B cells that have low affinity. Well, if there is a competition between the two, the cells with high affinity would bind the antigen better. And because those are the cells that are stimulated and kept as memory cell, the resulting antibody response would be higher affinity antibody from that cell. And therefore, the whole antibody response will be of higher affinity. Okay? And this is basically what this thing says. Okay? Imagine that you have two competing populations of people fighting for food. The strong ones and the weak ones, fighting for a limited amount of food, okay? And they're fighting each other. Obviously, the strong ones are going to win and competing for the food, and therefore they will survive, and therefore they will make kids who are also stronger than the other population. This happens molecularly at the immunoglobulin levels, in fact, in terms of affinity. So the other set of soluble components that are involved in immune response is a series of protein called complement. Okay, complement defines a number of different proteins, okay, that are generally inactive, but then when activated, they have a biochemical pathway that generates a whole bunch of different products, each of which has a specific immune function. Okay, and once again, I have to keep this very simplistic because it is a very complex mechanism. Okay, but the activation of complement proteins initiates a cascade which in turn either sequentially or at the same time turn on other proteins by cleaving various different products, resulting in active products. And the basic function of complement is they can lyse bacteria, that can actually puncture the wall of a bacterium and therefore killing it or destroying it. Also cells or viruses. They can opsonize. Again, they have coding properties that make the particle that is opsonized more visible to the phagocytic system. And they have a whole bunch of other properties, such as stimulation of various different inflammatory cells. So here is an example where you have a deposition of a complement component. Once again, I realize this is very simplistic. You may have a lot more in this than what I'm covering, but I'm restricted with time. And here is the formation of what is called a membrane attack complex, which is basically a chemical hole puncher, which in turn punctures the microbe membrane and then causes the membrane and the cell to lyse. Okay, and that is the result. Now, here is other components that are chemoattractants. I'll talk about chemotaxis in a minute, okay, that are chemoattractants, meaning that once activated, they call for other inflammatory cells to come to the site. Okay, they recruit other inflammatory cells, for instance, neutrophils or macrophages, to come to the site. This is kind of a tissue under attack, sending out an alarm for the soldiers to come in and defend it. Okay, and this is, I have these two boxes backwards, but anyway, this is the final, the structure of the microorganism, and this is the recruitment. And then, of course, you have opsonization. Other components simply adhere to a particular a cell, such as a microbe, and then a phagocytic cell, let's say a macrophages or a neutrophils, can recognize that particular component stuck to the microbe and then destroy it. So complement functions are varied and numerous. Okay, is this not working? So complement proteins, once again, are a whole bunch of different proteins, mainly produced by the liver. Okay, and what they do is they circulate as inactive molecules. Okay, however, once they're activated, for example, IgG bound to a particular microorganism, or IgM, exposes a site on the antibody molecules that is recognized by the complement proteins, and once they bind to that site, they get activated. Okay? There's a whole bunch of different products. The main ones are C1 to C9. This is just an example of how we name them. And there are two major complement pathways, okay, which are usually stimulated by different situations. The classical complement pathway usually is antibody-mediated, meaning antibodies such as IgG and IgM, once they are bound to an antigen, can activate the classical complement pathway. Okay? Now, on the other hand, the alternative or alternate pathway is a secondary pathway that, instead of being stimulated by antibodies, are stimulated by certain microbial components. Okay? And is indeed as part of the innate immune response. So here are the classical and alternate pathway. No, I'm not going to ask you to remember all these sequences. Okay? Just know that the final products of the complement cascade eventually converge into the final products that do whatever it is to be done. Okay? So the classical pathway, which is antibody mediated, can initiate a cascade of a whole bunch of different events that converge with C3 and then go down the line to all the other components. Or the alternative, or an alternate pathway, which has its own pathway and then converges here as C3, and then you goes down the line. And the bottom line, the message to take home here is either pathway results in the production of different proteins, which in turn have different functions for a particular event to be taken care of. Okay, and here's just another picture of one of the many sequence of events. In this case, it happens to be the generation of the membrane attack complex, which is once again a molecular hole puncher. And there's a whole bunch of events that follows a a sequential of activation of different components, cleaving of different products, and eventually you have the formation of a membrane attack complex, which in turn now at the very end punctures the membrane of the cell that has been chosen to be destroyed. Okay? Now, opsonization and phagocytosis, we went through this. Obviously, there are very important steps in getting rid of an insult or an attacking microorganism. So, microbes are either recognized by the classical or the alternate pathway, even depending whether they are hooked to an antibody or whether the alternate pathway is activated by the microbial components, okay? And then what happens? Some of these complement components are, again, opsonins, such as C3b, okay, which in turn now recruit various different phagocytes, okay? And also those phagocytes can also recognize antibodies attached to that particular microorganism. So the combination of the opsonins on the bug and the antibodies on the bug, okay, in concert can attract phagocytes, which in turn kill the bug. Here is just a opsonization picture where you have antibodies. This is a bacterium. You have antibodies attached to the bacterium, and now you have complement components, opsonins, attached to the bacterium, and now a phagocyte can recognize either the complement component on the microorganism or the antibodies on the microorganism, of both, because these phagocytes have receptors for these components, and now this gets internalized and gets destroyed. Okay, once again, opsonization meaning making a particle more visible, more appetizing to the phagocytic system. Now, the other thing that complement components do is getting rid of immune complexes. Immune complexes are basically a bunch of antibody and a bunch of soluble antigen, not particle, soluble antigens, that when stuck together, they form very, very large biochemical lattices, which in turn, sometimes they get so big, they can actually precipitate out a solution. Okay? And of course, the immune system wants to get rid of these complexes. Okay? Why? Because this is how the antibody is attached to that soluble antigen. Okay? And of course, you want to get rid of it. And they can create a lot of problems. Many autoimmune disorders are actually caused by the deposition of immune complexes. Once again, rheumatoid arthritis is a classical example. Okay? So the complement system tries to get rid of these immune complexes before they become so big that they become a problem. Okay? So here are basically the function of complement, which I want you to remember in broad category. Lysis, meaning the actual physical destruction of a cell membrane. Opsonization, okay, by opsonizing bacteria. Recruitment of inflammatory cells via chemotaxis, okay, and clearance of immune complexes. Those are the major basic function of complement. Okay, once again, simplistically put. Now, of course, as in any, and we'll talk about this when we talk about immunodeficiencies, okay, some of the immunodeficiencies are caused by defects in a complement system, okay? And there are many different components that may be defective within the complement system, and depending on where the defect is, you get a particular immunodeficiency. Whatever the phenotype may be, okay, you get a particular expression of clinical problems and pathology that is associated with that particular immunodeficiency. Okay? And of course, if your complement system doesn't work either partially or totally, then what happens? Well, you can't fight stuff as efficiently. So you have recurrent bacterial infection, you have deposition of immune complexes. Okay? CD3 deficiencies. If you are missing that particular component in which the two pathways converge, then the remaining pathway cannot be turned on. And of course, then the complement cascade cannot take its actions, and you have problems. Okay. Sometimes, believe it or not, even not turning off complement creates a problem, okay? Once complement is activated, you want to keep it in check, okay? Because you don't want it to go out of hand. So there are various different components, such as inhibitors, that once the action of complement is carried out, they turn it off. Well, sometimes there are conditions, and we'll talk again when we talk about immunodeficiency, in which failure of turning off creates a problem. And a classical example of this is hereditary angioedema, where you have the inhibitor of complements that are not turning off. Complement is activated continuously, and of course because some of the complement components increases capillary permeability, tissue edema, you have these kind of clinical pictures. We'll talk about that in a different lecture. Now, so we talk about humoral immunity, soluble components. What about cell-mediated immunity? Well, we talk about cell-mediated immunity, we basically talk about cells killing other cells. Okay, this is a picture of my brother-in-law during the first Gulf War. He was with a group of Rangers. We always tease him because of his mohawk. His whole platoon was like this. But basically this means these are the actual physical soldiers in the field that go out and do the job to kill the enemy. Okay, now, if you think cell-mediated immunity is not important, here is a situation in which you have a child with cutaneous candidiasis because he has a defective cell-mediated immune response. So, it can be pretty devastating. Now, cell-mediated immunity can either be T-cell dependent or T-cell independent. So in general, cell-mediated immune responses that tend to be secondary, okay, usually involve T cells activation. Once again, T cells being completely in control of the immune system once they have been instructed, okay? And we'll talk about the importance of these cells when we talk about HIV infection and AIDS. But you also have T cell independent cell-mediated immune responses, which do not rely, okay, at least initially, on the activation of T cells. And these are various different examples of T-cell independent: chemotaxis, phagocytosis, and the release of cytokines. Now, chemotaxis simply means the directed movement of leukocytes along the concentration gradient of chemoattractants. Okay? Now, what does that mean in English? Basically, the ability to detect an insult, okay, and to physically move toward that insult. And I'm going to use the analogy I used before, okay? What would you do if somebody's cooking lasagna or pizza on the floor here? You would smell the pizza, and you would physically move toward increasing concentration gradient of pizza smell. Okay? Now, as silly as it may sound, this is actually what cells do. They smell, to a certain extent, an insult by having receptors on their surface, noses, okay, that detect these smells, this chemoattractant, and are physically able to move toward where the chemoattractant smell gets stronger and stronger, just like you would move toward where the lasagna or where the pizza smell is getting stronger and stronger. Okay? Now, what are these smells? These smells are either derived from the microorganism themselves, or they are derived from the host calling for help. And what are the chemoattractants of the host? Various different cytokines, as well as complement components, which we just covered. Now, when you're dealing with, and of course, we talked about, let me go back for a minute. We talk about phagocytosis and the release of cytokines. Now, once again, cytokines are chemical mediators that are used by cells to communicate with each other. They're critical. Okay, when we talk about the immunodeficiency, I'll talk to you about David, the bubble boy, who was completely immunodeficient to the point that he had to live in a sterile bubble. Okay, cytokines were involved in that situation, and we'll talk about that. So these can also be released without initial activation of T cell. However, most cell-mediated immune responses are generated by activating T cells. The reason is helper T cells play a major role in all cell-mediated immune responses. Okay? And what they do, you see I have, okay, first of all, they direct the specificity of the response. Okay? T cells that are activated by recognizing an antigen that has been processed, by recognizing pieces of that antigen via their T cell receptor, those are the ones that now will instruct other cells to react against that specific antigen. So they direct the specificity. The other thing they do, and this is very important, they select the effector mechanism against the target cell, meaning that depending on what the antigen is, okay, you're not going to use the same weapons for the same enemy. Okay, you don't use a 9mm pistol against a tank, and you don't need to use a bazooka against a single individual. So the selection of mechanism is also achieved by helper T cell. Now, this is a very important point because unfortunately, there are situations in which this effector mechanism are poorly chosen or erroneously chosen, and what you have in immunopathology. Okay? In addition, they also push for the proliferation of appropriate effector cell type, meaning if you are going to choose an effector mechanism, you have to communicate and activate the cells that employ that effector mechanism. So helper T cells are critical in that too. And once again, if there are any aberrations in the communication between a helper T cell and the effector cell type, you got problems. Either too much or not enough response. Now, what are the effector mechanisms in cell-mediated immunity? Well, there's a whole bunch of different ones, okay? But these are classical examples. Cytotoxic T cells. Cytotoxic T cells are T cells that are the commando of the immune army. They physically go to a site where the target is, they attach to the target and kill the target. Or they could recognize a particular target because it's covered with antibody. Okay? Or even stronger, they can activate macrophage. We'll talk about activating macrophages in a few slides. So, again, this is a very important point because this will lead us into hypersensitivity and autoimmunity or even immunodeficiency. The decision of selection of which mechanism is critical for an efficient immune response. You gotta have the right weapons at the right time for the right situation. Okay? So once you get to the point that a cell is killing another cell, how does the killing cell kill the target cell? Okay? So this is obviously not a property only of cytotoxicity cells, but other cells of the lymphoid and even myeloid system. And again, I'm not going to get into great details, okay? But there are similar processes that are involved regardless of the type of cell that does the killing. Okay, so whatever it is that the cell that does the killing is, they follow a pattern that is similar to other cells that also do the killing. So there are three distinct phases. The first one is that the killing cell has to attach to the target. Now this is a critical point. This is hand-to-hand combat. Okay? This is not a sniper on the top of a building shooting the enemy across 1,000 yards. This is physical contact and combat between two physically attached cells. Once this particular relationship, physical relationship, has been achieved between a cell that does the killing and a cell that does the dying, then what happens is a calcium-dependent phase in which the killing cell, okay, discharges various different chemicals and other things that the cell keeps in vesicles, in the vicinity of the target cell, and then the late phase where the target cell is killed. Okay, those are common mechanisms to all different types of killing cells. So how do you physically now kill a cell? Okay, so now you have contact, you have chosen to kill the target cell. How do you kill another guy? Well, you can shoot him, you can, stab him with a knife, you can show him a picture of me in a Speedo, okay? Whatever it is, there are 3 examples in which you have target killing. Okay, the first one is the vesicles that I talk about contain a chemical called perforin, as well as various different polymerizing enzymes. And once the cell releases this chemical on top of the cell to be killed, the polymerizing enzyme, allow the perforin to polymerize and to puncture a hole in the target cell. Okay, so this is like perforin, enzymatic polymerization, the preparation of a membrane, and then actual physical polyperforming channels into the target cell. That's one mechanism. The other mechanism, instead of perforing, the target cell releases a whole bunch of bad crap, basically. Okay, in other words, digestive enzyme, reactive oxygen and nitrogen species onto the vicinity or on top of the target cell, and in this case the target cell gets damaged and dies. Now, in some cases, the actual killing cell dies too. There are certain cells such as neutrophils which extravasate a whole bunch of other stuff in trying to kill a particular antigen, and in doing so they also die themselves. And the last, and this is just a picture of the same thing. And then the last one is the actual secretion of various different cytokines. For instance, tumor necrosis factor alpha and beta, interferon gamma. Now these cytokines are used by the killing cell to stimulate apoptosis into the target cell. To actually trigger on a self-destructing mechanism on the target cell. And this, of course, takes a little bit longer because you have to have a little secretion of cytokine, this receipt of the cytokines from the cells to be killed, the activation of the cell to be killed, and the induction of apoptosis. And this is a picture of the same thing. Now, the last thing that I want to discuss is the activation of macrophages. Now, macrophages are very interesting cells in the immune system because they carry a lot of different hats. They do a lot of different jobs within the immune system. First of all, they're involved in non-specific responses because they're phagocytic cell. So like any other phagocytic cell, the first thing they do is they chew off the perceived enemy, for example, a microorganism. Now, because they're antigen-presenting cells, they're also turned on, they're also linked, just like dendritic cells, between the innate immune system and the acquired immune system. So they can turn the acquired immune system on as antigen-presenting cell. The other thing they can do is become actually effector cells themselves. Once the acquired immune system has been stimulated, T cells feed back to the macrophages, and depending on the level of activation of macrophages, macrophages can do a whole bunch of other things. So here is initial defense, but you have just simply getting rid of the microorganism, period. But the macrophage in turn can also secrete cytokines and instruct other cells, as well as present to a T helper cell. And then the T helper cell responds, feeds back to the macrophage, and now an activated macrophage, can do a lot of different other functions. And here's just some of the few things that a macrophage can do. For instance, it can turn on stimulation of pyrogens, turn on various different mediators such as prostaglandins, complement factor, Okay, it can select whether you go into a cell-mediated immune response or a humoral response. It can obviously activate lymphocytes via antigen presentation. It can act on the extracellular matrix. It can have microbiocidal activity. It can have tissue damage. We'll talk about that when we talk about autoimmunity and hypersensitivity. So the bottom line is it can have tumoricidal activity, antiviral activity. So the bottom line is once a macrophage is activated, depending on the signals that it receives from T cells, depending on the signals that it releases to other cells, and depending on the stage of activation, okay, you can have a lot of different results. Now, what's the problem with this is because, and I showed you in the very beginning the little critter with the machine gun, okay, because these cells are so powerful, okay, what happens is that cell-mediated immunity many times can create immunopathology, meaning that it actually can be the culprit in some pathological condition. And we'll talk about that when we talk about autoimmunity and hypersensitivity. Okay. So here is a pissed off macrophage. All right, if you have any question, if not, let's go eat.
When the cells secrete the performance, you said that they needed the calcium. How do they know, or how is the calcium on top of the target cell and not?
No, calcium